For everybody with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 3, Episode 61 of Surfing the NASH Tsunami, our somewhat abbreviated preview of a very exciting NASH-TAG 2023 conference, starts now.
Today, on Surfing the NASH Tsunami.
Every session has something that's going to smack people in the face. It's just that robust.
I guess at NASH-TAG in 2020, my comment was, boy, it felt like there was so much deeper to dive than y'all were diving at that point in time. And it's not 3 years later. This feels fantastically deeper.
Hope is such an important word, but hope is not a strategy. And what we can say today is we have something now, and there's real hope for our patients.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researcher and key opinion leader Dr. Stephen Harrison, liver wellness advocate Louise Campbell, and pricing and forecasting guru Roger Green as they preview NASH-TAG 2023 and discuss the release of Maestro-NASH Phase 3 trial results on today's edition of Surfing the NASH Tsunami.
Hi, it's Roger Green. Welcome to today's episode. It took us 3 weeks to find a day on Stephen Harrison's incredibly packed schedule when we could actually record the NASH-TAG preview episode. So, of course, when the recording date and time actually arrived, it was the day that Madrigal Pharmaceuticals released its Phase 3 Maestro-NASH data. Now, Stephen served as principal investigator, and the entire fatty liver world was going crazy. So we, in fact, recorded this session in the 1 hour gap after our Madrigal Roundtable conversation that served as the foundation for Surfing NASH Episode 60, and before Stephen rushed off to his next investor call. When you combine to this the challenges our intrepid engineer, Magic Mike Milson, had in returning from his Hawaii vacation, and some other commitments of mine, the entire schedule for this week has been thrown a tilt. This short but important conversation about Maestro-NASH and NASH-TAG originally was scheduled to post Wednesday evening, but we'll be posting Friday morning instead. Now, that said, I'm not sure that NASH-TAG needs a lot of promo. Based on what's going to come out about resmetirom and all the other exciting things that are going on, attending NASH-TAG in person or virtually should be a no-brainer for all of us. So listen to our conversation with Stephen, after which I'll be back for a brief business report. For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Today's episode was supposed to be entirely a preview of NASH-TAG, but because of this morning's events with Madrigal, and we have Stephen Harrison with us, we're going to spend a few minutes talking about the Madrigal release, and then about the NASH-TAG preview. And this should be a heck of an episode, even though it's just Stephen, Louise, and me. So, Stephen, congratulations. Banner day, huh?
Yeah, I really think so. You know, it's been a long day. I've been up really early this morning, kind of getting ready for the press release and the investor call that happened very early. If you're on the West Coast, I mean, my goodness, you guys were up at 5 o'clock in the morning listening. But I tried to think about what today means in the field of NASH, and I don't think there's a single word that can be used to describe where we are today. I would say it is a breath of fresh air for the field, and I used that word this morning on the investor call, and I think that is a clear understatement. When we embarked on developing resmetirom in the clinical setting back in 2015, 2016, I think we could all agree this beat our wildest expectations. It's taken a long time to get here. And if you look at the field of drug development in NASH and all the drugs that have fallen by the wayside and the challenges we've had with endpoint assessment, COVID playing a big role in the middle of this Phase 3 trial, just to be where we are today, I think, is monumental. It's landmark, it's Herculean, it's watershed, however you want to describe it. But who is going to benefit from this? That's the 25 million Americans, maybe more than 150 million patients worldwide suffering from this horrendous liver disease. And yes, while we still have a ways to go to get to approval, we've got the key ingredients needed to get there: safety, tolerability, and efficacy. So I want to just take a few minutes and just talk through a couple of those foundational items for drug development in NASH. And number one, if you look at the therapeutic index of a drug, there are two main components. There's efficacy and safety. And I would say based on the Maestro-NAFLD-1 data and more than 1,000 patients treated with 1 year of therapy, plus the extension trial ongoing, that there's enough data to say that this drug is safe and well tolerated. It's also oral once a day. So it's built for the long term, and that's what we need in these patients, something that is chronic therapy that can be given over several years if needed. So what are the adverse events that we're seeing? Well, again, recapitulating data from the Maestro-NAFLD-1 trial, the Maestro-NASH trial showed that the adverse events, particularly the serious adverse events, were balanced across groups. Among the adverse events, they tended to be GI in origin, with mainly mild intermittent loose stools to diarrhea and mild nausea. But in general, when you look at discontinuations for therapy, they ranged from 2 to 3% for 80 milligram in placebo to 7% for the high dose group. So in general, most people tolerated it very, very well and were able to stay on therapy. And you want a drug that really gets after the drivers of NASH. And I think resmetirom, being a thyroid hormone receptor beta agonist, it does that very well. It targets the toxic fatty acids that drive a lot of the inflammation and apoptosis, ballooning degeneration, and ultimately stellate cell activation. And so when we begin to look at the results where you hit both NASH resolution as measured by 0 for ballooning, 0.1 for inflammation, plus a 2-point improvement in the NAFLD activity score, and you're doing that about 10% of the time in placebo, and you're doing it 26% in the low dose and 30% in the high dose, giving you a treatment effect delta of 16 to 20% for both doses of drug, 80 and 100 respectively. That is impressive, particularly when you consider that in this modified intent to treat analysis of 955 patients, that 17% of people were excluded and counted as negative because they didn't undergo a follow-up liver biopsy. Also, biopsies done greater than 60 weeks outside of window, 52 weeks was the length of the study. Biopsies done 8 weeks or beyond after that because of COVID or whatever were also counted as null responders. So when you look at the data in total, you might say, why is the response rate lower? Well, you got to keep in mind in these phase 3 trials that if you didn't undergo the follow-up analysis, you were counted as a negative. So that's important. And then when we look at fibrosis improvement, again, reaching statistical significance, highly significant for both doses relative to placebo. You see a placebo response rate that was in line with the Regenerate trial and a drug response rate of around a treatment effect delta of around 12%. Again, in line with what we see with the beta-cholic acid. Importantly, we saw additional comments being made at the press release this morning that both endpoints, looking at the combined endpoint, not just an either or, but NASH resolution and fibrosis improvement was also hit. There was also a significance for patients having a 2-stage improvement in NASH. Now, these are top-line results. We don't have details about this. That will be forthcoming at additional liver meetings. But what also was important was a key secondary endpoint on LDL reduction that was statistically significant at both doses, and comments made by the CMO, Becky Taub, that there was also lipo-A, ApoB, and other atherogenic lipoproteins that moved in a very positive direction, suggesting an impact on cardiovascular risk over the long term. Granted, those trials haven't been done yet, and we don't have CV risk assessments, but we're moving lipids in the right direction. And so there were also a plethora of non-invasive tests that these trials have included. Just kudos to Madrigal for including almost every non-invasive test that could be utilized at the time this trial was initiated were included, from VCTE to MRE to wet biomarkers. And we see there will be more data forthcoming at congresses in relation to how those biomarkers did on treatment response, as well as hopefully outcome assessment. We also noted today that there was artificial intelligence data generated from both HistoIndex and PathAI, which I think is incredible. That data has not been evaluated yet, to my knowledge. It certainly wasn't presented today, but should be forthcoming. So in total, I think not only do we have very, very good histopathology data, we have good data on non-invasive test assessment, good data on LDL, but we also have a wealth of additional data that's forthcoming that we haven't even heard about today that I think will be incredibly helpful to the field as we move forward relative to non-invasive test assessment. Where does AI digital pathology fit in here, as well well as other assessments of lipoprotein. So it's a good day for NASH.
Stephen, fantastic day for NASH, and I think you're being a tad modest about your own role in all this. I'm mindful that when we started the podcast, the conversation that led to us doing this in the first place was about how do you handle biopsy in COVID and people shutting down trials and you saying, no, you don't have to do that. If you're thoughtful about what you put around it, you can keep going forward. And as I was listening this morning at 8 a.m. to the press statement, I thought back to that. And I thought back to, in fact, specifically what you wanted to do this podcast to do in the first place. Now, obviously, we've gone far afield from there, but that was the initial thought, was literally to educate people on that particular process. To this day, folks occasionally listen to that first episode when they're looking for guidance and tips on this stuff. I just think in terms of going full circle, I mean, I'm here because you were thinking about that stuff. And to hear that this morning and think about how all the pieces fit together was really just a fantastic thing.
Look, this is a team effort, right? I mean, so many people burned the midnight oil and just had a never give up mentality. That's just a statement on the team that really drove this to pull this through. You know, Madrigal deserves a lot of credit here.
Oh, a ton.
I think also Summit deserves a lot of credit. Summit, from a patient enrollment perspective, Summit was Herculean in enrolling this trial. And so kudos to that team as well for making that happen. But again, it was a combined effort. And what I've heard today from so many people is exactly what I wanted to hear. And that is, it's such an important day for our patients because hope is such an important word, but hope is not a strategy. And what we can say today is we have something now and there's real hope. There's real hope for our patients that they can just hang out and make it, work hard on lifestyle modification. You know, we have a drug that is going to be very helpful and meaningful to their health in the long term. So I'm just excited about that.
Stephen, just two more thoughts quickly, and then I want to turn to NASH-TAG. Number one is hope is not a strategy. It was something Peter Traber said on this very subject on the first episode of the podcast 2.5 years ago, almost 3 years ago. We're just keep coming back to episode 1. Number 2 is Donna Cryer couldn't make the podcast recording, which is later today, but she was able to do an interview with us earlier. And if you think of Donna as kind of the ultimate patient advocate, we asked Donna which group this meant the most to. And what she said first was investors, in part because if you want to talk about what gives hope to patients, it's the idea that the resources are going to be there to make all this real. And that one of the messages that she jumped on today, immediately, the first one she jumped on, was the message to investors that say you can trust this disease category.
Yeah, I think it's clear that what we had been missing from the puzzle of this is this a druggable disease? And I think we found out today that the answer is yes. So this should be, in my opinion, a rising tide that lifts all ships. And from Donna's perspective and what she's doing and all the great things that all patient advocacy groups are doing, is this gives just an incredible shot of energy and enthusiasm to disease awareness. There's going to be a huge drive to get out what NASH is to the population, not just doctors, but to the patients, and also how to diagnose it. I think we have a couple really nice algorithms from the AGA to ACE to the ASLD on how to diagnose these people, and I think that is going to roll out with authority, and I'm anxious to see that happen soon.
Excellent. So with that, why don't we shift gears? We have about a half hour left, 35 minutes to record. It's worth noting that resmetirom will be a significant topic of discussion at NASH-TAG as well, although hardly the only reason for people to go. But we'll skip the resmetirom discussions from NASH-TAG because I think people now will get that on their own. And if you can run us through the program and talk about what you think are the high points and the reasons that people will want to pay attention.
Sure. Well, we're back again starting on January the 6th, which is Friday. One of the events that I look forward to the most at this liver meeting is hearing the year in review. And Mike Charlton does the best job on the planet at summarizing data and giving us a good perspective. And that leads off this meeting, the year in review 2022. And boy, what a year it was. And I can't wait to hear how Michael puts that in context. And then we kind of mix things up a little bit. The first session after that is entitled Cover to Cover. And if you've been to prior NASH-TAGs, you'll remember that historically we lead off with just different drug mechanisms of action with pros and cons relative to what's working or not working. This year we decided to switch it up a little bit and we're doing what's called Cover to Cover. We're taking really 3 main drug classes and we're really doing a deep dive, not classes, but actually specific drugs that are in late stage drug development and just doing a deep dive into what we see. So those are resmetirom, obeticholic acid, and semaglutide. And I can't wait to hear what Vlad Ratsiu says about obeticholic acid and what Rohit Lumba says about semaglutide. I think week, and then I'll be speaking on resmetirom. The second session is latest clinical trial results, and this could have been the entire meeting. There's so many trial results that have come out, so many press releases that have happened. Vincent Wong, actually, we're honored to have him be a part of this year from the Asian PAC region. He's going to be summarizing all the data that was presented at AASLD. We're going to give him 20 minutes to do that, and then we're going to roll right into session 3, which is therapies in development. And we're going to go through a lot of what you guys have heard about recently: FGF21s, FASN inhibitors, acetyl-CoA carboxylase inhibitors, even DGAT inhibitors, some of the new siRNA data. What does that look like? Update on the PPARs, not just lanifibranor, but seraglitazar as well. And then we would be remiss if we didn't have something in here on foundational therapy, where we are with lifestyle, weight loss, and bariatric surgery. Man, there's been some incredible data that I feel like we've glossed over a little bit because of the drugs in development. But what a wonderful paper recently published in Nature Medicine on Fitbit, looking at thousands of patients and millions of patient hours worth of data on people using Fitbits, and the fact that 8,200 steps a day links to significant reductions in obesity and diabetes and depression, so many other things, PCOS, sleep apnea. So we want to hear about that. And then I think also going back a bit, we were really blessed this year to have over 40 abstracts submitted, many of these cutting edge abstracts. And as you guys know, if you've been to NASH-TAG, it's a very cozy setting. We like it that way. We want to keep it that way. And so we have limited space to present, limited time on the calendar for oral presentations, but we did select 10 oral presentations. So we're going to hear 10 really fascinating topics and give time for discussion for each of those. We'll have about 20 posters as well. Moving into session 4, what's happening in biomarkers and non-invasive tests. We're going to hear updates on Nimble and on Litmus from Arun Sanyal and Quentin Anstey. We're going to hear about non-invasive tests in general, and I've asked, we collectively asked Phil Newsome to come over from Birmingham in the UK to put it all in perspective. So the title of his talk is actually NITs for NASH, Making Sense of It All. Just kind of give us context. And then from there, we're going to drill down on that. Dr. Noureddin from Houston is going to talk about how to enrich NASH trials to detect that at-risk NASH population and hopefully mitigate screen fails moving forward in these trials. And then William Alizawi is going to talk to us about the clinical care pathway in NAFLD and how to use these NITs. And as always, great discussion, a panel discussion to follow. And guys, by the way, this is still the first day. Like, we're still on the first day. So session 5, advances in clinical trial design. Look, if you're interested in this field and you want to design a clinical trial, this is what you want to be at. Key caveats regarding combination therapy trials by Vlad Ratsiu. Key caveats regarding PNPLA3 HSD17 targeted trials by Rohit Lumba. I'm going to talk about caveats regarding cirrhotic trials. And we'll have a fantastic panel discussion. Becky Taub from Madrigal, Tom Capozzi from Intercept, Kathy O'Hare from GSK. Quentin and myself are going to discuss all of these topics. So what a great opportunity to hear what's going on. And that's the first day. Maybe I should stop there, take a deep breath, get some water, and see what you guys have to say.
And not only that, Stephen, but Jorn is joining us as we speak. Jorn, how are you?
Good afternoon or good evening. I'm fine, thank you. Sorry for being so late. Good to see you, Stephen.
Jorn, Stephen just did day 1 of NASH-TAG in a breathtaking 12 minutes and then asked for questions right as you were popping in. So while Jorn is catching up and Louise is catching up, my first comment is this. The last session you're talking about really strikes me as being particularly fascinating, and it speaks to how far we've come in terms of what we're willing to look at. If you remember when we talked, I guess, at NASH-TAG in 2020, my comment was, boy, it felt like there was so much deeper to dive than y'all were diving at that point in time, and it's now 3 years later, I guess. And this feels fantastically deeper. I mean, you're looking at things at very, very basic levels that make sense, and then pulling those in a clinical trial and practical application just strikes me as, well, okay, resmetirom results for a sea change. 2 sea changes makes an ocean change, or whatever the heck it makes. It feels like that, just vast.
Yeah, I think there's been a lot of, I'm hoping that there will be a lot of wind added to the sail of NASH drug development with what we're seeing today. I've had conversations with different sponsors today who are excited for Madrigal. I think we all should be excited for Madrigal, even if you're trying to develop your own asset. But look, there's room for improvement, right? A 20% treatment effect delta on NASH and a 12% for fibrosis, leaves room for improvement here. There's an opportunity for injectables, an opportunity for orals, and an opportunity to combine some of these agents. I mean, we're seeing compounds that modulate de novo lipogenesis that are at least non-invasively having a huge impact. To me, I think that's an opportunity to combine with something like a THR beta that we could call foundational. A structurally engineered fatty acid, I think, would fit in very nicely with this compound as well. Those are both, whether you look at a FASN inhibitor, oral, once a day, well tolerated, CFA, oral once a day, well tolerated. Is there an opportunity to put those 3 or 2 together in a combination trial, multiple combinations, fixed drug combinations? Do we do injectable for induction followed by oral maintenance therapy? There's all kinds of ways to look at this. The other thing in combination trials or other trials that need to be designed, and I'm talking about the one I'm doing on cirrhosis, why do we only have one adaptive trial? Why do we only have one trial looking at prevention of progression of disease and everything else looking at regression of disease? I mean, to me, these are like conversations I have with my son about why do we park on driveways and drive in parkways? Why are apartments so close together? Why do we call them apartments if they're so close together? Why do trucks carry shipments and ships carry cargo? I mean, we can make that argument about why is drug development the way it is. I mean, let's throw it on its head, take the lessons learned and move forward. So I think
that's almost an English lesson for me, Stephen. I like those analogies. If I look at day one program, the exciting thing is you cover all the MOAs, and I'm not sure we're ready to move in combination for all these. Theoretically in academic medicine, it's fascinating to discuss about them, but I think there's so much more we got to learn about the monotherapy. And looking at day one program, you guys really did a great job in putting them all together. I mean, they're all there, and that's, I think, the reason why I'm looking forward to go to NASH-TAG, just to hear, an update on that.
Yeah, thank you. And I think you being present will also be very helpful because of the grasp that you have on this field and the literature, and you're providing feedback. I mean, I think that's another one of the really exciting things about this meeting is it's such a cozy environment. People feel like they can speak their mind and they can say what they want to say and they can ask the provocative question, and you're very good at that. So you being there will be very, very helpful.
Amen to that. Stephen, I think that last thing you did about cargo and trains and all that might end up being our first ever publicity poster. I just think that's brilliant. And the way that just rolled off your tongue.
I had a couple of thoughts. You might have to reorder the first session, starting with resmetirom may not be the best place because once you get the open mic, you're never getting away from it. So you might have to put it further at the third one of the Ugly Sisters. You just also, with today's announcement, blown session 4, because if I was Phil Newsom, Marzin, or William, I'd be really hacked off if I'd already done my slides on the way that we can now rewrite pathways because of the breadth of non-invasive technologies that were used in this trial. That to me was the groundbreaker today, is the fact that I can see pathways. I can see how they can be developed. It's something we can physically start to work on now to develop those networks. And you've just made every single patient organisation for NAFLD and NASH very, very busy. But yeah, if I'd done my slides, if I'm any of those 3 guys, I'm really hacked off now because I might have to write a few more.
They better get to work now because it's important to get the latest and greatest there, and I think we will.
You know what? If they have to work a little bit over the holidays, what a reward they're going to get when they show up in Deer Valley. What a beautiful place and a great venue to have these type of discussions.
Yeah, I confess I'm jealous. I can't be there. I shouldn't be there for reasons I can't be there, but boy, do I want to be there. It's just going to be such a great event. We'll be covering it, but we'll be from afar. Some of us will be from afar. Day 2.
Well, day 2, I have to confess, it's my favorite day because it's also my 54th birthday. So we're going to be having a party on Saturday, January the 7th. And so how do you beat the first day? Well, we got to try. So we're going to jump into session 6, which is heterogeneity of disease and the impact on clinical trial outcomes. And we're going to look at those topics, Jorn, that we discussed at Dublin and Paris. What are some of these factors that are driving the outcomes we're seeing in clinical trials, good, bad, or ugly? Genetic influences, microbiome, cryptogenic alcohol use and abuse, endocrine influences, and then we're going to have a panel discussion. Again, fantastic lineup of speakers that are going to take us through this, but probably here, the biggest amount of panel discussion we've built into the program is this session outside of the fireside chat at the end, because I do think this is what we're talking about, or what are these influences that are driving some of the outputs that we're seeing in these clinical trials. And maybe the biggest one to me is the surreptitious alcohol use. Are we really doing a good job of quantifying, qualifying how much people are drinking? And should we be building in things like phosphatidylcholine or PEth testing? I think that's really important. Now, there are arguments about the right cutoff. What should we use? And I think we need to have a robust discussion about this role of genetics. I mean, should we be genotyping all of our patients to know how they're likely to respond or not respond in clinical trials? So that's the first session. And then jumping beyond that, we're going to talk about what's new and how to interpret. And again, another topic that's been huge in 2022 is this whole nomenclature process, NAFLD versus MAFLD versus something else. And so we're going to hear from Mary Ranella. Of course, Phil Newsom is there as well to speak to this, to speak into us wisdom about what's happened, about where we are today, and what still needs to be done utilizing this Delphi process. And I don't think that we could have picked two better people to chair this than Mary Ranella and Phil Newsom because they're so open to listening to different viewpoints from around the world and from our patients and everybody. And I've just really been impressed with the way that they've taken on this challenge and tried to make it all inclusive. So we'll hear where we are with that. Then another exciting topic that I know you also are interested in is this whole imaging liver and spleen volume change that we're seeing with some of these therapies and what that means, not only to interpreting histopathology, but what it can mean to an outcome measure. So we'll hear a little bit about that. I also want to bring in a little bit here on visceral adipose tissue quantification, what we call VAT. We know that links to an outcome. DEXA scans can do this very easily. Should we be building this assessment into our clinical trials? Predicting therapeutic response with NITs, I'm going to give that talk. I feel very passionate about that. I did a little bit of that at EASL back in London in June. We've got some updates for you 6 months later. I'm going to share that with you. And then Rohit Lumba is going to talk on predicting outcomes with NITs. And then Naeem Al-Khoury is going to give us an update on digital pathology. Where have we come? Where do we still need to go? And how do we take the data we have today and apply the AI path paradigm to that? And again, a robust discussion on all of this, from spleen volume, liver volume, to the context of use of predicting response to therapy, outcomes to therapy with NITs, and digital path.
That's like the next generation NITs here, right? Because, we're moving clearly beyond the diagnostic biomarker field. It's not, according to the best criteria, not to diagnose the disease, but how to predict the response, which, as you said, if we have 30% or a delta of 20% of effect size, do I want to keep the other 80% on drug if it's not working? That's maybe the next, level question. But we need these biomarkers. And I think, therefore, that seventh session for me is really, I agree, that's a highlight of that day.
Yeah, and actually, I'm going to give you a little snippet. If you're listening to this podcast, you will hear this. Nobody else will. One of the oral abstracts is looking at the lanifibranor data and predictors of non-response. So it turns out that if you don't drop your ALT at all at week 4, you don't respond. Now, that sounds simple, but that's a predictive response. We're going to see more and more of that. So thanks for highlighting that.
And I can share, I gave an update presentation on that. I'm happy to, I'll be seeing what viewpoint you're going to be taking, but the therapeutic response with NITs is clearly, the big question is, what do we do with those between 17, which predicts outcome, and 0, which doesn't predict outcome, right? So that's the subclass of patients then with the ALT response drop. But anyways, I think that's leading too far at this point.
That's okay. We don't want to get out over our skis because we're in Deer Valley. We don't want to get too far out over our skis. But I do think we need to have a discussion. The other part of that is the 17 unit per liter actually, is that agnostic to mechanism, or does it depend on the mechanism of drug that we're treating? So we'll have to talk about that too. But
one of the interesting things to me about your ALT Lani story is it really changes the economics of Lani, right? Because if you can start to pick off fit patients that will fail at 4 weeks, then we're not any longer telling people, hey, you got to stick with this therapy 6 months to figure out what you got, which is better for the patient because if it's not working, they can get treated faster. And frankly, it's better for everyone in the healthcare system because you can save a bunch of money.
That's right. Yeah, that's a great point. The economics of this will become more and more important as we get closer and closer to our first drug at CVS, Walgreens, or wherever. Look, we're going to come back at 4 o'clock and we're going to have session 8, which we have entitled Potpourri. And here we're going to have more oral presentations, more oral abstract presentations, but we're also going to mix it up a little bit. That's why it's Potpourri. We're going to have endobariatric management of liver disease. This is going to be a wonderful presentation. I can't wait to hear that. And then we'll have Scott Friedman, who is literally the father of the stellate cell. Been on the podcast, big fan of the podcast. We're a big fan of him. He's going to tell us the year in review, the top 5 papers in basic science in NASH. I can't wait to hear what he pulls up and discusses.
And in his end of year review, he already complained that he'll have to do this. I think that work still needs to be done on his side. But anyways, I'll be looking forward to it.
Actually, he told us, Stephen, he shared with us some of what he's going to do, not all of it. But then he said he's got analogies and metaphors that we're going to have to stay tuned for. I'm wondering if he's going to try to get Michael Charlton to run for his money. It'll be really exciting to see how that talk turns out.
I think so. It's almost like bookends, right? We start with Michael and end with Scott here on year in review. And then we have Dilly. And who better to give Dilly in the setting of NASH than Naga Chalasani? We talk about, Scott being the father of the stellate cell. Many people would consider Naga the goat of Dilly. I mean, he has spent a career focused on this area, and it's great to have him be a part of this. Not that others don't do a lot of work in the field either, but I really respect the work that Dr. Chalasani has done in helping me design clinical trials in NASH and ensure that we are appropriately evaluating for drug-induced liver injury here. And then finally, I would say, for me, the highlight every year is session 9, this regulatory landscape fireside chat. And I just cannot wait to share a lot of the discussion that came out of the Mosaic meeting that happened 2 weeks ago at the French Embassy in Washington, where we were fortunate enough to have 3 different divisions of the FDA present. We had cardiologists, nephrologists, radiologists, hepatologists. We had biostatisticians present. We had the 3 editors from the leading journals, Nature, Lancet, New England Journal present, and had a robust discussion. And I'm going to bring that discussion into this session and extend it. And we have industry sponsors. We have patient representatives here. It is going to be a robust discussion that we have gotten used to having. And I think you will be remiss if you do not show up to hear this. It's always a wonderful end to a great 2-day session. And we're going to wrap it up. And that's it. We're going to go off into 2023 and do great things.
Exhale. That's a tremendous amount to do in 2 days. Now that you've done all that, do us a favor. Tell us the 2 things that you didn't mention because as you flew through that are just going to smack people in the face or have the potential to smack people in the face.
Well, there's so much here, Roger. I think the things that are going to smack people in the face are some of the discussions around the NIT field and where we're headed and some of the comments that Jorn was alluding to. That's a hot topic, the whole non-invasive field. And then I think the other one is going to be advances in clinical trial design. I think there's just been some amazing work being done here. Having said that, I could think of 2 or 3 others that are going to smack people in the face. So I think every session has something that's going to smack people in the face. It's just that robust, I think, personally.
I'm intrigued by the co-founders session, if you'd like. We're discussing alcohol, some of the genetics. We all lump this up in the big phase 3 trials and still get a signal. That's a sign for me that the underlying disease is modifiable and the number of response or the response rates we're getting here is all impacted by that. If you then come better at quantifying that, I think the metrics might change a little bit in terms of, placebo response and treatment response. That is interesting to learn how much of the underlying alcohol they're consuming, which is moderate, maybe light, might be influencing those outcomes. So I think that's interesting. It's going to be a speculative talk. I don't think we have a lot of data. Maybe we can generate that at one point when we actually use drugs.
Well, there was an interesting session that Roger and I both attended at EASL, and it was was on using hair follicles to measure alcohol because it stays in the system, and you got a longitudinal review of somebody's alcohol intake. That's obviously just giving up alcohol a week before you were to enroll in a study. So Roger and I were discussing it at the time, sitting in the room, but actually it now potentially comes into the fore as a potentially better marker than some of the others because of its longitudinal review. Throw that one into the mix. But it looks like a great session, and I'm certainly going to be looking forward to session 7. And on that point, do you think the discussion around nomenclature now, given the press release today, does that take a different slant on it about naming and renaming a disease during the process of the approvals of drugs in this time frame?
As long as we don't change the definition, it's okay. From my perspective, my biggest fear is that we have to go back and redo studies because we changed the definition of the disease. I don't mind changing the name as long as we don't change what we're defining, because I think at that point we have to not only change our drug development, but we have to change all the non-invasive tests that we've worked on to this date. So look, if you want to call it something other than NAFLD or NASH, that's fine. Just don't affect the definition.
It's interesting because some of the feedback we've gotten on that is exactly as you say. And some of it is, in fact, Jeff Lazarus was saying this morning, that one of the concerns that he's hearing is that at the point in time where we're going to start educating patients about what's going on, we're going to be giving them a name they don't know yet. And his comment, which I thought was interesting, was that all that work's got to get done and really start to get delivered before we have drugs. Because once we start educating people about how to use drugs, they better know exactly what the disease is, and it better be the name that's going to stick. Because at that point, patients will get confused, which is interesting because I hadn't thought about that particularly before this morning. And then it came up with Jeff and came up again with Donna. So obviously, people who are at the back end of the process, that's something that they're thinking about a great deal. Louise, anything you want to add?
I'm just looking forward to it, and I love the fireside chat.
We'll be doing follow-on sessions each day. What we did last year was we learned that we could get together for breakfast or get people together at breakfast time for a while to talk about the episode from the day before. We will be doing that again so that we can get some coverage on what's going on. Hopefully, Stephen will be able to join us one of those mornings, which he was able to do last year, and Jorn will be able to join us one or both. I know Louise will be there for both. I'll be there for both, and we have other folks who volunteered to come talk with us in the mornings. Scott Freeman, Quentin, and Steve are two that immediately come to mind. So we're looking forward to just a great meeting. And as I say, I'll be sad not to be there in person, but very, very excited to see how it goes down because it sounds like the mind's playground, I got to tell you.
Well, thank you. I hope it lives up to the expectations that we've built for the meeting, and can't wait to talk about it on the back end.
Well, I will be doing that for a while, so that's exciting. Okay, so Stephen, thanks for joining us today. Congratulations. I know you've got more to do today than we can imagine. We'll be touching base with you in Park City, I hope. Okay.
Sounds good. Good to see you guys.
Good seeing you, Stephen.
And now for the Season 3, Episode 61 Business Report.
Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to heroes. Two thoughts on this. First, for those of you who saw Volodymyr Zelensky's address to the U.S. Congress earlier this week, the moment when he said Slava Ukraini and the audience replied Heroyam Slava brought a huge smile to my face and a slight glisten to my eye. Looking at the pictures of President Zelensky, I may not have been the only one with that reaction. One other thought. I take it as heartening on a number of levels that our download audiences in major Russian cities have stayed strong over the past 10 months. In fact, podstatus.com tells us today that we are among the top 120 medical podcast downloads in Russia. That's the first time we've been in the top 200 in over a month. I think all this is good.
This is our last live episode of the year.
Today and over the weekend, we will drop conversations and snippets from this episode and Wednesday's Maestro Nash Roundtable. On Monday, December 26th, we'll drop our next year-end episode, this one featuring interviews with Mazen Noureddin, Ken Kuzi, and Jeff Lazarus, with the individual conversations posted in full on Tuesday and Wednesday. The final episode of the year will drop Thursday, December 29th, featuring Stephen Harrison in an interview actually recorded before the Maestro Nash data dropped, Naeem Al-Khoury and Ian Rowe.
This may also be our last thanks for listening statement of 2022.
For whatever reason, our listener numbers were a bit light the last couple of weeks, but the last 2 days have approached the higher levels we've seen throughout the year. This next set of conversation and year-end interviews are each fantastic, each unique, and you'll probably want to hear them all at least once. Thanks in advance for your interest and getting this year to numbers we didn't even imagine at the outset in terms of downloads and interest. And with that, I'm off for today. Thanks to Jake and Magic Mike, who've been heroic this week and will continue to be so as we wrap up the rest of the year, and Eric and Steve for all you guys do. And to the rest of you, our listeners, to whom I thank the most and for whom we do this, enjoy this episode, check back next week for the year-end pieces, and whatever the weather has in store for you this week, stay safe, surf on. We'll see you on the podcast. Bye-bye now.
Please join us again on Monday, December 26th, when Mazen Nuruddin, Ken Kuzi, and Jeff Lazarus join Yarn, Louise, and Roger to discuss their own perspectives on 2022 in fatty liver disease on a special year-end episode of Surfing the NASH Tsunami.